Hello, welcome to this presentation on the plant vaccines. In this particular presentation, we will see how the plant vaccines are being produced and how we can utilize these plant vaccines to combat the current and upcoming pandemics like SARS-CoV-2 and others possible pandemic which might invade the human population in coming decade or in a coming time. So, firstly, we will talk about what is the plant vaccine. So, according to the World Health Organization, the definition of vaccine is given as such. A vaccine is any preparation intended to produce immunity to a disease by stimulating the production of antibodies. This definition was given by WHO in 2018. A vaccine could possess purified microbial toxins, biomolecules, killed or attenuated microorganisms, recombinant vectors. Any vaccine possesses two basic immunological characteristics. One is specificity and another is memory. First vaccine is reported in 1796 when Edward Jenner attempted vaccination for smallpox in an 8-year boy named James. Louis Pasteur named these attenuated strains as vaccines, where Vacca refers as cow because the first vaccine was made from the cowpox fluid. Now we will talk about plant various types of the vaccines. So first we will talk about the live attenuated vaccine. A live attenuated vaccine is a live microbial preparation in which microbes have been weakened by growing them on non-specific host. Attenuated vaccines produces similar immune response as wild strain, however doesn't cause disease. It produces a strong immune response that often lasts lifelong. Example for such live attenuated vaccines are influenza and MMR, when MMR represents the three vaccines together which are given as a single dose or together in a child's. MMR stands for measles, mumps and rubella vaccines. Another one is the inactivated vaccines. Inactivated vaccines is a killed microbial preparation in which microbes have been killed by chemical or physical treatment. These vaccines are safer and more stable than attenuated vaccines. An inactivated vaccine provides low immunity, therefore it is needed to be administered in multiple doses. Example for such vaccines are polio vaccines and rabies vaccines. Next one type is subunit vaccines. The subunit vaccines do not use the entire microorganisms. In its place, these vaccines use only the antigens that can cause or induce immune system to activate. These vaccines can be prepared by purifying the desired antigen from the pathogenic organism or by genetic engineering or gene coding. The vaccine protein of interest is inserted into a vector and subsequently expressed in a host system. These vaccines are safer and possess minimum side effects due to the use of a specific gene. Example for such vaccines are hepatitis B, human papilloma virus. Also, one thing I would like to tell about the hepatitis B, that India is the very prominent country where the hepatitis B vaccines are produced massively and being exported to the worldwide. Main companies producing the hepatitis B vaccines are the Bharat Biotech and Serum Institute of India, which is located at Pune. Another sort of the vaccines are known as toxoid, a disease caused by microbial toxins instead of a microorganism itself, can be cured by toxoid. A toxoid vaccine is an inactive form of microbial toxins. The inactivation can be achieved by the treatment of toxin by chemicals such as formaldehyde. Example of such vaccines are tetanus and diphtheria. Diphtheria is again a respiratory disease which is caused by a bacteria named as Corynebacterium diphtheriae. Another sort of the vaccines are the conjugate vaccines. Conjugate vaccines are the variation of subunit vaccines. This vaccine was developed to provide immunization against certain microorganisms that possess polysaccharide layer around them. This layer protects microorganisms from the host immune system. In conjugate vaccine, antigenic proteins are linked with microbial polysaccharides so that immune system can produce immunity against it. This polysaccharide attachment helps the immune system to defend the body by reacting with the polysaccharide coating of pathogen. Example for such vaccines are HIV vaccine. HIV stands for the Haemophilus influenza B, and another example for this is meningococcal vaccines. Another sort of vaccines are known as the DNA vaccine. This is very recent technology. In this technique, gene encoding for microbial antigens is directly introduced into host body. This microbial gene controls the host cell to produce the antigen protein of interest. Expression of microbial protein in host cells result in the specific immune response in host cells. Example for DNA vaccine is the Zydus Sedela, which is effective against the SARS-CoV-2. Another sort of vaccines are the recombinant vector vaccines. Recombinant vector vaccines are functionally similar to DNA vaccines, but attenuated virus or bacteria are used to introduce the microbial DNA into the host cells. Vector term is used for the attenuated virus or bacteria that used to carry the microbial DNA within a host. Example for these sort of the vaccines are the VSV, vesicular stomatitis virus, and CMV, cytomegaloviruses. Plant vaccines is the very recent vaccines which have been developed uh, by the extensive effort of the biologists with the medicinal experts. In the early 1990s, a novel approach to produce edible vaccine in plants emerged. Plant vaccines are a type of subunit vaccines and commonly called as edible vaccines. Plant-derived vaccines are less toxic and cheaper than the conventional vaccines. The oral activity and heat stability of these vaccines make them an obvious choice for poor countries. Now we will talk about how the production of plant vaccine takes place. So the firstly Mason and Ernst describe the detailed procedure for the production of plant vaccines. Plant vaccines are produced by the molecular forming, which is also known as forming. Plant vaccine is produced by the inserting a microbial gene which is encoding for the microbial protein into a plant. Expression of this microbial gene in plant cells will produce the protein that can be purified or consumed along the plant tissue. Hence, it is an advanced biological technique used for the production of pharmaceutically beneficial proteins in plants. In this flow chart, we will see that the uh, important gene of interest which can be incorporated within a plant by two means, via plasmid vector or by a viral uh, phages. And by the plasmid vector we will genetic transformation and regeneration in plant will happen, and these plants will be known as the transgenic plants. While with the infection of viruses, the gene can be incorporated within the plant genome, and these both, if the gene of interest is expressed within that plant, then the effective antigens or antibodies will be formed within a plant that can be extracted or can be used as raw plant materials, and these will be known as the edible plant vaccines. Production of plant-based vaccines requires the following steps. First one will be the selection of gene. The gene encoding the antigenic proteins that stimulate the immune system should be selected for the process. The requisitions of gene include its ability to assemble in virus or plasmid, to survive in GIT, which stands for the gastrointestinal tract, and also to trigger the oral and mucosal immune responses. Second step will be the selection of a specific plant. A plant selected for the plant vaccine production should be easily available and must be the one that can grow easily in the local region. The plant should be easy to transform and needs to express high quantity of protein. In addition, the plant must not contain any toxic compound within it. An edible plant is most preferred for both human and animal application. Currently, many plants have been used for the production of plant vaccines such as tobacco, potato, tomato, bananas, and other cereals plants and alfalfa plant also. Another step is insertion of gene in a plant. The microbial gene can be transferred in plant genome by two methods. The first one is stable genomic interaction. The structural gene, or which is also known as antigenic gene, is loaded on a plant transformation vector. The vector is introduced into plant cell using Acrobacterium tumefaciens, chromosomal integration, or by microprojectile bombardment techniques. Another way of inserting the gene of interest within a plant genome is the transient expression using the viral vectors. The microbial DNA encoding for epitope is inserting in a plant pathogenic virus. This is genetically engineered virus infects the plant cells and start replicating there. Along with the viral gene, the microbial gene of interest also can be expressed and produce protein that gets accumulated inside the plant cells. The last step in the plant vaccine production will be the harvesting. Expression of the microbial proteins occurs in various parts of plants. The plant tissue can be consumed directly or after cooking. Alternatively, the protein can be purified by the downstream processing and subsequently used in the developing tablets, capsules, and other pharmaceutical products. Generally, plant vaccines are considered safe. However, it is important to check the potential and toxicity of product in animal models before being used commercially. Now we will talk about how the plant vaccines are affecting, or what is the methodology or mechanism of plant vaccines, how they function. So here will be the steps involved by in this diagram. So here we see that plant vaccines which are taken by our oral consumption, and these plant vaccines will go into the gastrointestinal tract, and there these uh, other tissue of plant will be digested, and these antigens or plantigens will be released within the intestinal tract, and these there are the M cells within the intestine which will absorb these plantigens, and they will pass these plantigens to the plantigen representing cells, which is known as APC or PPC. Activation of T and B lymphocyte will occur, and production of immunoglobulin A antibody will happen, and that will neutralize that particular plantigen which we infested or eaten. A plant vaccine administered orally stimulates the mucosal and humoral immune system of the body. Mucosal immune system is the primary defense system and an appropriate site of vaccination. After the administration of plant vaccine, it stimulates the immune system in a systemic manner. This will be whole mechanism of these plant vaccines will be having three steps. First will be the entry of antigen. The antigen present in a plant cell can be consumed in form of raw plant products such as fruits, vegetables, and through the tough outer cell wall that can be protecting antigen from degradation by the gastric enzymes, which will be attacking on that when the particular plant vaccine is in our gastrointestinal tract. And breaks to release the antigen of intestine will be stopped by these hard or tough outer cell wall of these plants. Another step is the absorption of antigen. Free antigen present in intestine are absorbed by M cells, which I already explained in the previous diagrams, and that will be passed to the follicle associated epithelium of Pierce patch in the intestinal lining are present over the Pierce patches in the ileum. Ileum is a part of intestine, and the gut associated lymphoid tissue will be absorbing that. Once these plantigens or antigens are absorbed, then immune response will occur. M cells pass the antigens to the macrophages and other antigen presenting cells with further present the antigen to local T and B lymphocytes. These activates B cells migrate towards the mucosal associated lymphoid tissue mold, and B lymphocytes differentiate into plasma cells and synthesize immunoglobulin A antibodies at the mucosal surfaces that neutralizes the antigens in the intestine. In addition, few antigens taken up by intestinal dendritic cells DCS, and that further includes the production of immunoglobulin G antibodies, which will impart the systematic immunity also. Now we will talk about the potential of this plant vaccine by their effect on the bacteria, viruses and other parasites. So first we will talk about the plant vaccine which work against bacterial infections. So pathogenic bacteria cause a variety of infections such as tetanus, typhoid fever, diphtheria, syphilis, cholera, foodborne illness, leprosy and tuberculosis. And tuberculosis ranks second the world's leading disease which caused the million of deaths worldwide. In every in year 2012, 8.6 million cases were reported. Out of that, 1.3 million deaths were reported due to the tuberculosis. This report was published by WHO in 2013. Streptococcus, group B streptococcus is another frequent cause of life-threatening infection during the first 2 months of life. Food and waterborne bacteria such as Salmonella typhi and Campylobacter are responsible for a recent drastic increase in diarrhea cases. Even there are lots of the medicinal literatures which reveals that the increasing rapid increase of the diarrhea is giving the problem. Even the probiotics which are taken during that diarrhea, they are also not being that sufficient to treat the diarrhea. So these are the few challenges which can be overcome by these plant vaccines. The discovery of new organism and new strain of many familiar bacteria present in a study challenge to the researchers toward the development of microbial control measures. The application of conventional vaccine has controlled the death rate of viral diseases and bacterial diseases. However, the development of plant vaccine could possibly provide a support to the conventional vaccines. So here we will see that few very dangerous bacterial infections which are being treated by the vaccines produced from the plant-based materials. So first one is the enterotoxigenic bacteria E. coli, and for that the plant plant-based vaccines which was made by using the enterotoxin uh, surface protein, or what you can see labile enterotoxin B subunit protein, which was produced in various plants like tobacco, potato, and maize kernels that was successful for treating these enterotoxigenic attacks or problems caused by this enterotoxigenic E. coli. Another one is the Vibrio cholera, which causes the disease cholera. For that, also the plant vaccines were made by using the cholera toxin Bu subunit, and this was produced in the potato and tobacco, tomato and rice plants. So there are a number of the plant-based vaccines were made to combat these uh, bacterial infections, and that were successful. Still, we hope that in upcoming decade we will be excel in this field and to treat any sort of the bacterial diseases. As we know, the most of the bacterial diseases which were traditionally treated by the uh, antibacterial drugs, but as we know that bacteria has the adaptive immune system, and they easily can get the antibiotic resistant or multi-drug resistant. So in that case, if we are able to produce the plant vaccines which are highly effective against the bacterial infections, that will be a good effort or good achievement of the whole scientific community across the world. Now we will talk about the plant vaccines which worked against the some viral infections, or we will say the some deadly viral infections. Viruses cause various dreadful diseases in humans and animals. Most of the epidemic diseases are caused by the viruses because of their ability to spread so fast. Viral infections are difficult to treat because they grow inside the host cell and quickly adapt to a new environment by making changes in structure. Human immunodeficiency virus, AIDS, hepatitis virus, rabies virus, variola virus, polio virus, and Japanese encephalitis virus are some of the most common viral pathogens. These viral pathogens cause severe infection in humans and resulted in several deaths worldwide. The application of conventional vaccine has controlled the death rate of viral diseases. However, we need some more development in the plant vaccines so that we can combat any sort of the viral infection coming by using the plant vaccines alone. Some viral pathogens like hepatitis B virus, whose surface proteins were incorporated in plant as plantigens, and we formed or produced the plant vaccines known as plantigens in the plant of tobacco, potato, carrot and banana and tomato. And for the HIV type 1 virus also, we provided the gene which code for the V3 loop of GP120 protein and peptide of V3 loop of GP120 protein and TAT protein in the tobacco and spines plants to produce plant-based vaccines. Similarly, with that, rotavirus, variola virus, Japanese encephalitis, Lysa virus, human papilloma virus, and rabies, for that also the appropriate antigens producing gene of interest was incorporated within the plant genome, and that resulted in the formation of that plantigens based plant vaccines. Another plant vaccines which are very effective against the parasites. So in tropical and subtropical regions of the world, parasitic infections are leading causes of diseases and subsequent deaths. Malaria and lymphatic filariasis and toxoplasmosis are the most common and severe parasite diseases. Malaria is an endemic disease occurring in almost 108 countries of the world and kills a large number of people every year, which is given data by the WHO in 2016. Nearly 30 million people in the United States are affected with the toxoplasmosis, according to the report of the Central Center for Disease Control and Prevention (CDC). which was published in 2017, about 120 million people are infected by lymphatic filariasis worldwide. 65% of them are present in the Southeast Asia group. Development of edible vaccines to prevent the parasitic infections would provide an alternative to be existing prophylaxis and treatment methods. At present, some parasitic proteins are expressed in proteins for the preparation of edible vaccines. Some edible vaccines in tobacco plant were produced for treating the malaria, which is caused by the plasmodium uh, falciparum, plasmodium vivax, and other diseases like toxoplasmosis, which is caused by the toxoplasma gondii. The recent use of the plant vaccines can be utilized to treat the SARS-CoV-2. So plant-based vaccines for COVID-19 can be developed either by expressing the antigens, which those vaccines will be known as plantigens, components for the SARS-CoV-2 for including active immunity, or expressing the antibody, which is known as plant antibodies against the virus to provide a passive protection. Plant-based vaccines are considered as third generation vaccines. The production approach of a plant-based vaccine involves the cloning of vaccine candidate into a plant expression systems, which is capable of prompting the expression of the candidate gene in plant, which then produces the antigenic or protective proteins. So as we know, coronavirus are enveloped positive sense RNA viruses and are characterized by the club-like spikes that project from their surface, an unusually large RNA genome, and unique replication strategy. Coronavirus cause a variety of diseases in mammals and birds, ranging from the enteritis in cows and pigs, upper respiratory diseases in chickens, to potentially lethal human respiratory infections. Coronavirus are the largest group of virus belonging to the nidovirales order, which include family Coronaviridae, Artiviridae, Mesoniviridae, and Roniviridae families. So novel coronavirus which cause the disease name as the COVID-19, and this COVID-19 stands for the coronavirus disease 2019, which was originated from the Wuhan in China, and this disease is caused by the virus which comes from the family of SARS-CoV-2. SARS stands for the severe acute respiratory syndrome. So this COVID-19 is a respiratory disease which is very lethal, and the mutations within this virus ranging from the alpha, beta, gamma, delta, and currently Omicron variants are reported, which are lethal to the most of the human populations, and delta variant was reported the most dangerous mutation till the date, which caused the millions of deaths uh, in the in last few months, or we will say in last 1 year. So how we can produce the plant vaccine against COVID-19? So there are some efforts to produce the plant vaccine against COVID-19, and the Serum Institute of India has announced it has partnered with the UK biotech company known as Spy Biotech, a spin-off the University of Oxford, to develop a COVID-19 virus-like particle VLP based plant vaccine. The vaccine has entered in first or second phase in Australia. VLP or virus like protein resemble the SARS-CoV-2 virus, the VLP is constructed incorporating all key structure proteins mirroring the molecule structure of coronavirus. However, it doesn't cause infection as it doesn't have the genetic material within it, and it replicates like a natural virus. VLPs can be produced using mammalian cells, insect cells, bacteria, yeast or plant cells. Of the five COVID-19 VLP vaccines, virus-like proteins in clinical trials listed by World Health Organization, one plant-based VLP vaccine is in third clinical trial. Medicargo developed this CoVLP, COVID virus-like protein vaccine within 20 days after once they received the genetic sequence of SARS-CoV-2. To design a VLP, the company selected the spike proteins for SARS-CoV-2, the most widely used target antigen for COVID-19 vaccines. Among the structural envelope protein, membrane protein, and nucleocapsid protein because of its high immunogenicity and crucial role in viral cell entry. Medicargo's first products, quadrivalent virus-like protein vaccine for seasonal influenza, completed the third phase clinical trial in 2020, was the first plant-derived human vaccine that was well tolerated and provided substantial protection from the influenza virus in adults. So as we have seen that in the year of 2020, the Medicargo has successfully produced a plant vaccine for the deadly disease influenza, and to take knowledge from there, we can be sure that in maybe in recent coming days there might be we might get one plant vaccine which may be working to treating the SARS-CoV-2 disease or COVID-19. In the trial, we found that 3.76, 7.5 and 15 microgram of COVID virus-like protein with or without ASO3 or CpG 108 proteins were intramuscularly injected in a 18 to 55 years old at interval of 20 days in a random manner and particularly blinded clinical trials. The immunogenicity resulted demonstrated that that adjuvant formulations have greater potential to improve humoral and cellular immune responses to the COVID virus-like protein vaccines compared with the non-adjuvant formulations. All subjects receiving the adjuvant vaccine developing neutralizing the antibodies as the immune response after the second dose in all plant groups. Also, here we should talk about what can be the challenges of plant-based vaccine against the COVID-19. So, as we know that this SARS-CoV-2, these are the viruses which are very rampant mutating viruses. So, though they will be facing the same problem, the plant vaccines have the same challenges as the other vaccines which we have right now, like Covaxin, Covishield, and other vaccines like Pfizer. The same problem will be faced by the plant vaccines. As the virus mutates, the efficacy of the vaccines can be terminated or can be reduced, or sometimes it may be ineffective. So the same thing should be taken in consideration, that when we are designing the plant-based vaccines, we should we should take lots of the trials that that particular vaccine should be having the potential use for not only the current mutations of the COVID-19, but also it can be the effective for the all the predicted mutations which may come in SARS-CoV-2 new strain of the viruses. So that was from my side, and I will close the presentation with this. Thank you so much.